हेलो फ्रेंड्स 16 नंबर का हमारा चैप्टर है एक्सक्रेटरी प्रोडक्ट्स एंड देयर एलिमिनेशन। स्टार्ट करते हैं। एनिमल्स एक्यूमुलेट अमोनिया, यूरिया, यूरिक एसिड, कार्बन डाइऑक्साइड, वाटर एंड आयंस लाइक सोडियम आयन, पोटेशियम आयन, क्लोरीन आयन, phosphate, sulfate, etc. either by metabolic activity or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous bases waste. Ammonia, urea, and uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with the minimum loss of water. The process of excreting ammonia in ammonotalism, many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surfaces or through or gill surfaces in fish. As ammonium ions, kidney do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conversion of water. Mammals, many terrestrial amphibians, and marine fishes mainly excrete urea and are called urotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain the destroyed osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with the minimum loss of water and are called uricotelic animals. And the survey of animal kingdom presents a variety of excretory structures in most of the invertebrates. These structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidneys. Some of the structures are mentioned here. Protonephridia or flame cells called flame cells are the excretory structures in platyhelminthes platforms. Example planaria, rotifers, some annelids, and the cephalochordata. Amphioxus regulation, that is osmoregulation. Nephridia are the tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous wastes and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustaceans like prawns. Human excretory system. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, a urinary bladder, and the urethra. Figure 16.1. Kidneys are reddish brown bean shaped structures situated between the levels of last thoracic and third lumbar and the abdominal cavity. Each kidney of an adult a human measures 10 to 12 centimeter in length, 5 to 7 centimeter in width, 2 to 3 centimeter in thickness, with an average weight of 1 to 20 to 120 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which an ureter blood is a broad funnel shaped space called the renal pelvis, is which with projections called the calyces, and the one layer of kidney is tough capsule. Inside the kidney there are two zones, the outer cortex and the inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyx. The cortex extends in between the medullary pyramids as renal columns called columns of Bartini, figure 16.2. Each kidney has nearly 1 million complex tubular structures called nephrons, figure 16.3, which are the functional units. Such each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of renal artery. Blood from the glomerulus is carried away by an afferent arteriole. The renal tubule begins with a double wall cup-like structure called Bowman's capsule, which encloses the glomerulus along with Bowman's capsule is called the Malpighian body or renal capsule or renal corpuscle, figure 16.4. The tubule continues further to form a highly called network, proximal convoluted tubule PCT. A hairpin shaped Henle's loop is the next part of the tubule, which has descending and ascending limb. The ascending limb continues as another high is called tubular region, called the distal convoluted tubule (DCT), and DCTs of many nephrons are open into a state which into a straight tube called a collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscle (PCT) and DCT of the nephron are situated in the cortical regions of the kidney, whereas the loop of Henle dips into the medulla. In a majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called as extra medullary nephrons. Now, the efferent arteriole emerging from the glomerulus forms fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion. And they are glomerular. That takes place in different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus and is called the glomerular filtration. On an average, 1100 to 1200 milliliter of blood is filtrated by the kidneys per minute, which constitute roughly one-fifth of the blood pumped out by each ventricle of the heart in a minute. Glomerular capillary blood assure causes filtration of blood through three layers, that is the endothelium or glomerular blood vessels. The two layers on the epithelium cells of Bowman's capsule called podocytes are arranged in an intricate manner. Intricate manner since the since to life some minute spaces called filtration slits or slit pores. Blood is filtrated to finally through these membranes that almost all the constituents of the plasma except the proteins pass on to the lumen of the Dauman's capsule, and therefore it is considered as a process of ultrafiltration. The amount of the filtrate formed by the kidneys per minute is called glomerular filtration rate (GFR). GFR in a healthy individual is approximately 125 milliliter per minute, that is 180 liters per day. And the kidneys have are built in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus JGA. JGA is a special sensitive region formed by cellular modifications in the distal convoluted tubule and the afferent arteriole at the location of their contact. A fall in GFR can activate the JG cells and JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. The comparison of the volume of the filtrate formed per day 180 liters per day with that of the urine released 1.5 liter suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called a reabsorption. The tubular epithelium cells in different segments of nephron perform this either by active or passive mechanisms. For example, substances like glucose, amino acids, sodium ion, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous wastes are absorbed by passive transport. Passive transport. Reabsorption of water also occurs passively in the initial passive transport. Reabsorption of water also occurs passively in the initial segments of the nephron, figure 16.5. During the urine formation, the tubular cells secrete the substances like hydrogen ion, potassium ion, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid base balance of body fluids. Function of the tubules. Proximal convoluted tubule (PCT). PCT is lined by simple cuboidal brush border epithelium which increases the surface area of reabsorption. Nearly all of the essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ion and ammonia into the filtrate and by absorption of the HCO3 negative from it endless loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The ascending limb of loop of Henle is permeable to water but almost impermeable electrolyte. This impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentration filtrate pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Distal convoluted tubule DCT. Conditional reabsorption of sodium ion and water take place in this segment. DCT, distal convoluted tubule, is also capable of reabsorption of HCO3 negative and selective resecretion of hydrogen and potassium ions and NH3 to maintain the pH and sodium potassium balance in blood. collecting duct. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Large amounts of water could be reabsorbed from the region to produce a concentrated urine. This segment allows passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of H positive and sodium ions. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentration urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite directions and thus forms a countercurrent. The flow of blood through the two limbs of vasa recta is also in a countercurrent pattern. The proximity between the Henle's loop and vasa recta, as well as the countercurrent in them, help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmol per liter in the cortex to about 1200 milliosmo per liter in the inner medulla. The gradient is mainly caused by NaCl and urea. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, some amounts of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. The above described transport of substance facilitated by the spatial arrangement of Henle's loop and vasa recta is called the counter current mechanism. Is called the counter current mechanism. Figure 16.6. This mechanism helps to maintain the concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate urine. Human kidneys can produce urine near 4 times concentrated than the initial filtrate formed. Regulation of kidney function. The functioning of kidneys is efficiently monitored and regulated by hormonal feedback mechanisms involving the hypothalamus, JGA, and to a certain extent the heart. Osmoreceptors in the body are activated by changes in blood volume, blood body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate these receptors, which stimulate the hypothalamus to release antidiuretic hormone ADH or vasopressin from the neurohypophysis. ADH facilitates water reabsorption from later parts of the tubule, and thereby preventing diuresis and increase in body fluid volume. You can switch off the osmoreceptors and suppress the ADH and release to complete the feedback of ADH. Can also affect the kidney function by its constrictory effect on the blood vessels. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby GFR. The GG juxtaglomerular apparatus plays a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR can activate the JG cells to release further to angio angiotensin second being a powerful vasoconstrictor increases the glomerular blood pressure and thereby aldosterone causes reabsorption of sodium ion and water from the distal parts of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin and angiotensin mechanism. An increase in blood flow of the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause dilation of blood vessels and thereby decrease the blood pressure. ANF mechanism therefore acts as a check on the renin angiotensin mechanism micturition. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system CNS. This signal is initiated by the stretching of the urinary bladder as it gets filled up with urine. In response, the stretch receptors on the walls of the bladder send signals to the CNS. The CNS passes on motor messages to initiate the contraction of smooth muscles um, of bladder and simultaneous relaxation of of the urethral sphincter, causing the release of urine. The process of release of urine is called micturition, and the neural mechanism causing it is called the micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic pH 6 and has a characteristic odor. Now, on an average, 20 to 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of main metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, urea, and histone bodies, ketonuria, and the urine are indicative of diabetes mellitus. A role of other organs in excretion other than the kidney, lungs, liver, and kidney also help in the elimination of excretory waste. Our lungs remove large amounts of CO2, approximately 200 milliliter per minute, and also significant quantities of water every day. Liver and largest gland in our body secretes bile containing substances like bilium, bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with the digestive waste. The sweat and sebaceous glands in the skin can eliminate certain substances through their secretions. Sweets produced by the sweat glands is a watery fluid containing NaCl, small amounts of urea, lactic acid, etc. Through the primary function of sweat is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the waste mentioned above. Sebaceous glands eliminate certain substances like steroids, hydrocarbons, and waxes through sebum. This secretion provides the protective, oily covering of the skin. Do you know that small amounts of nitrogenous waste could be eliminated through saliva too? Disorders of the excretory system, malfunctioning of kidneys can lead to accumulation of urea in blood, and a condition called uremia, which is highly harmful to may lead to kidney failure. In such patients, urea can be removed by a process and hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into a dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysis unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid, dialysis fluid having the same composition as that of plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on concentration gradient. As nitrogenous wastes are absent in the dialysing fluid, and these substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the body through a vein after adding antiheparin to it. This method is a boon for thousands of uremic patients all over the world. Kidney transplantation in the ultimate method of correction of acute renal failure, kidney failure. A functioning kidney is used in transplantation from the done from the from the donor, preferably a close relative, to minimize its changes of rejection by the immune system of the host. Modern clinical procedures have reduced the success rate of such a complicated technique. Renal calculi, a stone or insoluble mass of crystallized salts, oxalates, etc., formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney, excretion and their elimination, excretory products and their elimination. Summary: many nitrogen containing substances, ions, CO2, water, etc., that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretion vary among animals, mainly depending on the habitat. The availability of water, ammonia, urea, and uric acid are the major nitrogenous wastes excreted. Nephridia, Malpighian tubules, green glands, and the kidneys are the common excretory organs in animals. They do not only eliminate nitrogenous waste but also help in the maintenance of ionic and acid base balance of body fluids. In humans, the excretory system consists of one pair of kidneys, a pair of ureters, the urinary bladder, and urethra. Each kidney has over a million tubular structures called nephrons. Nephron is the functional unit of kidney and has two portions: arterioles, fine branches of renal artery. The renal tubule starts with the double-walled Bowman's capsule and is further differentiated into the proximal convoluted tubule (PCT), Henle's loop HL, and distal convoluted tubule (DCT). And DCTs of many nephrons join to a common collecting duct, many of which ultimately now open into the renal pelvis and through the medullary pyramids. The Bowman's capsule includes the glomerulus to form malphizion or renal corpuscle. Urine formation involves three main processes, that is filtration, deabsorption, and secretion. Filtration is a non-selective process performed by the glomerulars using the glomerular capillary blood pressure. About 1200 milliliter of blood is filtered by the glomerulus per minute to form 125 milliliter of filtrate in the Bowman's capsule per minute. GFR GG, a specialized portion of the nephrons, plays a significant role in the regulation of GFR. Nearly 99% of reabsorption of the filtrate takes place through different parts of the nephrons. PCT is the major site of reabsorption and selective secretion. AH12 primarily helps to maintain osmolar gradient 300 milliosmo per liter 1000 to 1200 milliosmo per liter with the kidney interstitium DCT and collecting duct allow extensive reabsorption of water and certain electrolytes which help in osmoregulation. H positive, K positive, and NH3 could be secreted into the filtrate by the tubules to maintain the ionic balance of pH of body fluids. The counter current mechanism operates between the two limbs of the loop of Henle and those of vasa recta capillary parallel to Henle's loop. The filtrate gets concentrated as it moves down on the descending limb, but it's diluted by the ascending limb. Electrolytes and urea are retained in the interstitium by this arrangement. DCT and collecting duct concentrate and filtrate by about 4 times, that is from 300 milliosmo per liter and 1200 milliosmo per liter. They are an excellent mechanism of conservation of water. Urine is stored in the urinary bladder till the voluntary signal from CNS carries out its release through urethra, that is that is mixed nutrition. Skin, lungs, and liver also assist in the excretion. Excretory and products में हमने देखा पहले से. Human excretory system, then urine formation, then function of the tubules, mechanism of concentration of the filtrate, regulation of kidney function, micturition, role of other organs in excretion, disorders of the excretory system, then summary. So thank you so much for watching. Please like, share, and subscribe.